Good afternoon, one and all. Myself, Dr.A.Pauldasan, working as an assistant professor in the Department of Botany, Bishop Heber College, Trichy. Today, we'll discuss about and we'll get knowledge on plant tissue culture, its definition, history, as well as advantage and application. The first, we'll discuss about the definition. There are two types of tissue cultures. One is plant tissue culture as well as animal tissue culture. The tissue culture means a process or technique of making plant tissue grown in a culture medium outside the organism. Any tissues which is taken from the plant body of animal or plant, it has been cultured in a artificial medium in a controlled environment is termed as tissue culture. Two types of tissue culture is there. One is animal tissue culture, the other one is plant tissue culture. As a department, as a botanist, we are focusing only on the plant tissue culture. Plant tissue culture is a technique of used to maintain, grow plant cell, tissue or organ under sterile condition on a nutrient medium, which means any part from the plant. Example, cells, any cells, tissues, simple tissues or complex tissues, parenchymatous cells, any tissues or organs or parts of the flowers, or parts of the plant. Anything which is taken from the plant is grown in a sterile condition. Sterile condition in the sense of without any contamination, it's in a controlled environment. A nutrient in medium, suppose we have to grow a plant in the laboratory condition, the plant needs some nutrients. That nutrients we have to provide in the culture condition. The other name for plant tissue culture is micropropagation. Micropropagation means using a very small piece of plant, very small size of plant or size of plant parts is termed as micropropagation. Two fundamentals or two concepts are essential in plant tissue culture. The first one is totipotency. Totipotency means the ability of a cell or ability of a tissue or ability of a plant organ has the potential to divide and redivide and divides and make them into a plant organ or plant parts. The other one is dedifferentiation. Dedifferentiation is a process which tissues that undergo cell differentiation. The cell which is taken from the plant, it has to undergo divisions and it has to undergo divisions again and again and it produces a primordial cell. The last one is competency. Competency means cell retain ability to grow or dedifferentiate. Any cells which has the potential, any cell which have the capability of grown into a new organ. The three fundamentals, totipotency, dedifferentiation and competency is very essential in plant tissue culture. And the history, Heberland is the father of plant tissue culture. Heberland is the first person demonstrated the concept and delivered the concept of plant tissue culture. He used four parts. First one is from leaves, petiole, granular hairs and as well as stamen hairs. He tried the four different parts of the plants and utilized that explant and grown explant in a crop nutrient solution. At the end of his experiment, he says that, to my knowledge, no systematically organized attempts to culture isolated vegetative cells from higher plants in simple nutritive solutions that have been made. So he is the first person, Heberland is the first person demonstrated, explained the concept of tissue culture. His research work has been published in Vansika Academy of Science in Berlin 1902. This is a small pictorial image of plant tissue cultures. The first image is the granular hairs. It produces the callus. It's a enlarged. The image B is the enlarged version of a callus, and from the callus, shoots are arising. And D is only the shoots has been isolated and shoots has been removed from the callus. It has been cultured in a separate medium and it's rooted plant, and the rooted plant is hardened. Different persons after the Heberland, different persons have worked on the tissue culture. In 1902, he has proposed the in vitro culture. In 1922, Robinson successfully cultured root and stem tips, and 1939, Gerbert, White and Nobocart established the endless proliferation of callus culture. So these three persons is the student of Heberland. The Gerbert, White and Nobocart worked on the plant cell and they proposed the concept of callus culture. So this is the first callus which has been worked by Gerbert, White and Nobocott. In India in 1957 only, the two persons, Guga and Maheshwari, started working on plant tissue culture in the University of Delhi in the Department of Botany. Guga and Maheshwari also demonstrated, also proposed the concept of culturing a plant from the anthers. So anthers can be used as a explant to form a new plant. Advantages, the plant tissue culture helps to mass propagate the plant. Very small piece of plant, very minimum piece of plant is utilized to make a many number of plants. From a single plant to thousands or lakhs of plants we can able to produce. If you want to develop a disease-free plants, tissue culture helps to develop a disease-free plants, and plant tissue culture helps to conserve the plant. Conservation of endangered plant or rare or endemic plant. So tissue culture helps continuous supply of a plant to the industry. So throughout the year we can develop the plant. The tissue culture process doesn't depend on the environmental conditions or something. Identical plants, we can produce a particular character from a plant. Embryo rescue. Embryo rescue means the embryo cannot able to develop a plant. The embryo is viable, but due to some reasons, due to some external forces or due to some environmental changes, the embryo cannot able to germinate. That embryo can be rescued from the seed. It has been cultured in the plant nutrient medium that can grow in an individual plant. In the horticulture, some changes, some morphological changes we can induce through the tissue culture. The last one is in vitro pollination for breeding purposes. So we won't depend on the hybridization in the field condition. With the help of tissue culture, we can hybrid the plants. The applications, four major sectors, four major regions are there. The plant tissue culture has been utilized. The first one is agriculture, forestry and pharmaceutical. The last one is genetic engineering. We'll go one by one. The first one is agriculture. In all the sectors, in all the regions of agriculture, they are utilizing the techniques of plant tissue culture. The tissue culture allows the production and propagation of genetically homogenized disease-free plant. So with the help of plant tissue culture, we can produce the disease-free plant. Another one is it is used to induce the somoclonal variation. Somoclonal variation means somatic clonal variation. The variation which is seen in the somatic cells. And genetic variability induced by tissue culture. So we can induce some sort of genetic variability in the DNA segments. And in vitro regeneration, mass propagation techniques, genes transmitted is in 3 species. If you like to transfer any genes from one plant to other plant, tissue culture is the base. Without the tissue culture process, it's not at all possible to transfer one gene to other gene. And the tissue culture use in vitro culture of immature or immature zygote embryos. As like the embryo rescue techniques, some of the zygote is not able or doesn't have the capability to regenerate into a plant. So that immature zygote can also retrieve from the plant. It has been cultured. That immature zygotic embryo helps in development of intergeneric crosses. The last applications in agriculture is it helps to break the seed dormancy. Some seeds can germinate within a month, or some seed can germinate even after 6. Some seed can germinate even after 1 year. That can be broken. That seed dormancy can break in the tissue culture process, and vitality of the seeds to can able to check the viability of the seed. In the forestry regions, the plant has been the tissue culture helps in the forestry sector, like to conserve the endangered plants, to conserve the endemic plants. Any parts, any plant parts, any tree which is very rare, which is very limited in population, that can be propagated through the tissue culture process. And another one is cryopreservation. Cryopreservation means storing a small piece of plant in a liquid nitrogen. So cryopreservation means cryo storing the plant cell or plant pot under -196 degree liquid nitrogen. Genetic transformation. So for any genetic transformation, if you like to produce GMOs, genetically modified organism, the organism which is transferred to the plant, one organism to other plant, the intermediate technology is the plant tissue culture. Without plant tissue culture, it is not at all possible to transfer one gene from the other gene. So that is one of the important applications in plant tissue culture. The last one is pharmaceutical industries. Many pharmaceutical industries are depending on the in vitro techniques. So it's not all possible to get into the field every time to collect a plant and to collect the extract from a single plant. It's a very time taking process. It's a very laboratories process. So with the help of tissue culture techniques, we can use the tissue culture techniques to produce a secondary metabolites. So the first image, the first one is the single cell has been cultured. It has been propagated in the techniques and it induced the callus. From the callus, it produces the hairy root culture. So hairy root culture is some chemical components which always depends on the depends in the roots. That root culture has been used. So the callus has been cultured in a cell suspension culture. Cell suspension culture means growing a plant cell in a liquid medium, not using the solidified medium, not using the solid medium. The callus has been cultured in a liquid medium. It's a cell suspension culture. From the cell, the secondary metabolites are produced with the help of bioreactors. Many of the pharmaceutical industries, many of the drugs has been which is taken from the plant through the cell suspension culture. This is the image of cell suspension culture. So here is the example, few examples of plants and its chemical components, the type of culture. The last one is hairy root culture. Through the tissue culture process, through the in vitro propagation techniques, the plant pot, any plant, leaf or shoot or axillary bud, anything can able to produce the root. So some metabolites are occurring in the roots. So it's not possible to dig the root of the plant from a ground. It's very time taking process. So what they will use is they will use the plant tissue culture technique. They can choose any plant, any part from the plant. They cultured in an artificial medium with the plant growth regulators. They can induce the root. That root has been cultured in a liquid medium. From that root, the secondary metabolites has been excreted. So here the few biological properties, few chemicals has been isolated from the plants. So here is the end. I summarize. So we seen that the definition, history, as well as the advantages and applications of plant tissue culture. Thank you.